All right, so welcome everybody. So I think this is the first seminar of the series for 2023, and it's a great pleasure for me to introduce Professor Stephan Huckemann and Professor Kanti Mardia. I will try to be as brief as possible in introduction. So Professor Huckemann is received his first degree in mathematics from University of Giessen in Germany. Then he visited many other places, including University of Michigan Ann Arbor, ETH. He was at Saudi Academia for a while. Then he went back he went to University of Paderborn, still in Germany, and many other after that. He got his habilitation at the University of Göttingen, when I believe he's still, when I believe he works now, and was awarded a DFG Heisenberg Research Fellowship. He continued at the IMS at University of Göttingen, and now he holds the new chair of Non-Euclidean Statistics. So his research includes statistical shape analysis and many applications, in particular to biomedical sciences, and many other things. Professor Kanti Mardia is studied in Bombay for his master and for bachelor and master in statistics. Then at University of Pune for his master in pure mathematics. He moved for his PhD to University of Rajasthan. Then he went to Newcastle upon Tyne. He had the academic position at the Institute of Science, Mumbai, University of Hull. Many other places he visited. He currently has a joint appointment, I believe, between University of Leeds and Oxford, and also I think the Indian Institute of Management, Ahmedabad. So I can say many things. He has written tons of scholar articles and books. I have some. Here I have one. Yeah, another one is in the office. I read it on a daily basis in practice. So he's an expert in multivariate statistics, statistics of manifold, statistical shape analysis, many other things. He received the Guy Medal in silver by the Royal Statistical Society, Wilkes Memorial Award from the American Statistical Association. He has been awarded a lifetime achievement by the International Indian Statistical Association, a Mahatma Gandhi Gold Medal of honor by the NRI Institute. He has been given the 40th Fisher Memorial Lecture at Oxford, the Mathematical Institute in Oxford. He was very recently appointed as Officer of the Order of the British Empire. This is news of a few days ago, I believe. So, well, it's an honor, a great pleasure to introduce both of you, and we are looking forward to your seminar. Thanks a lot for being here.
Okay, shall I start now?
Yes, please.
Okay. First of all, let me apologize, Sandro and Kathleen, that I kept on changing my mind. So it has been a lot of statistical fluctuations. And I'm grateful that you kindly agreed as we thought, look at our slide and looked at the audience. So eventually I started to think that better if I give introduction. So my introduction would be statistical, and the more profound would come from my co-speaker Stephan, not only statistics, but also the applications. Mine is very dry in the sense of only focusing on main thing, what Stephan is going to do on the statistics. And he's going to touch so many areas that only I can give you a glimpse as much as possible, and I won't take much time. So I will move to the next slide. So various things are going to come. Actually, I'm just giving you overview so that you start getting a bit hungry what's going to come. So the first is the thread would be of principal component analysis. Most of you, of course, will be familiar, but just to recall that eigenvectors give useful dimensionality reduction And so to play a game, there is a toy example. Most of my will be toys, so that, but they are serious in sense, gives glimpse the depth of what statistics can do. So here is this data, if you can, sorry, I've gone, I think it is, so I've gone now, I have to go back, but it's going further. So sorry, I think that will happen. This is quite a regular thing I do. So you can see the data all in 3D, but how to summarize? One way would be to have this eigenvector PCA, principal component, so that this red one is a serious one. So if you want only one-dimensional summary, then you will go for red. If you want 2 dimensions from 3 dimensions, then you will go to red and green both. So these are the two, some representation visual that how it will look when you rotate, and which I'm not going to do. Okay, I think it will. And these, some of the slides, I think it is cut off, but are from Steve Maron. I borrowed those slides from him. So next one, continue with PCA. I think the world started changing. People started getting into manifold, and they found life is not as simple as what Hotling had at his own time. So that life became a bit complicated. People started working on several data, and you will see some data later on, or you will know of some data yourself. So there are 4 points, and then how to get what is PCA would mean. And one way is to find some form of best small circle. And here is one which you have. So this, I think the work is just about 10 years ago. So it is in some sense relatively bottling in this. I think there is a time difference. I think there's a lot of time difference that tools are now coming, I think, because people started using linear whenever they could, and now they've woken up. The linear statistics sometimes gives all stupid answer. So then we come to the second innovation, that from sphere, how to go to a torus. This is 2-dimensional representation. The data in a cross section, it is planar representation. You can see red dots and other dots. And then the way to view perhaps is a donut, which gives you help in visualizing, not to eat just now, but it's not transferable. So that's a donut, and it is a great Stephan who has done, and his group, I think, has done the real work, and I was just an instrument in it. So then torus PCA, which is what came from sphere, and what great thing was done that somehow they deformed torus data onto sphere and use sphere, just now what you saw PCA, and key step is how to do it in some serious way, some keeping topological structure. So this is cutting open along a circle. So you have got 2 circles, you separate, collapsing each circle to a point, and then they come to the key step that you have to restore the cyclic topology, you join together. So that's the key idea. But for my simple mind, I kept on, asking Stephan and his group that it's too tedious for me, I think. So my simple way is torus in 2D, how you go to sphere in 3D, and then you can do PCA. So simple, there are 2 angles, for torus, 0 to 2 pi, I can understand. And then most of you will know spherical polar coordinates. So there's a theta and phi, 2 angle. But you will know that co-latitude or latitude pi by 2 minus theta goes from 0 to pi, and that goes to 0 to 2 pi. So simple-minded way of looking at it, that this x2 is a user-friendly. It can go on a sphere without any botheration. But this X1, how to bring it in simple-minded way is just divide by 2. However, this is absolutely watered down way of looking at it, because there are too many tedious things. Singularity comes, and how to cope with it, and where to cut a circle where you saw donut was cut, and so there is quite a lot of very difficult points which have been has been done. So then that's the one thing, and then PCA on one side, and then we come to cluster analysis. You are familiar, cluster goes reverse way of PCA, that you are given unknown group. So here are 3, celebrated Fisher data. And then there is one way to do it, and this is from our multivariate book, which is the second edition, which is still with Wiley, so that you can do multivariate normal mixture. But then one of the problem, and most of you will know, how many groups and how, what way, there are many ways, what are the tuning parameters. So the tuning parameters play a key role because it is not model based in general. I think the clustering is what is called unsupervised learning. So it is not an easy. So what has happened that if you have 3 component, it goes like this, 4 you have like this, and then if you do BIC, selection, then G becomes of this form. So this gives you, again, a glimpse of what cluster analysis is, although it becomes tedious when Stephan, I think the life is not easy on torus. I wish it was. So then we have got another thing will come, shape and size and shape. So this was the book Alessandro was just showing, I think. He was flicking that cover page. So the shape took also time to come into forefront. And David Kendall and Fred Bookstein, they are the 2 pioneers who did work to describe mathematically. So shape is the description of object after ignoring changes in location, scale, and rotation. So location, if I move my head backward, forward, I'm relocating my head. Scale, I think because of zoom, I'm coming nearer and nearer. And rotation, if I do like this, I think I'm rotating my head. And I hope I don't do full spherical rotation, the neck will go back. So the key point, the way it is, the subject I developed is the landmarks, some are anorthomical, and we are working on some called SMILE project. So I've taken lips, so there are 10 landmarks, and they've been properly located, and there's a big story. So that happens when I move my lips like this, and you do spread it out corner to corner, smile very badly, or become sad or whatever. So it keeps on recording this. But in size and shape, the location and rotation are allowed, but the size, like male and female lips, their size is different, which one has to take into account what we are working on. We have got controls of 2 types. So that gives you idea of size and shape, and which you will see effectively, Stephan, when he goes into the more harder problem. So this is just an overview, and I'm not selling these books, but just to remind myself, basically, that I've written something, directional statistics, where I said angles, and when I said sphere, are here in directional statistics. There are other books, of course. When I said about size and shape, and is it in this book? And then when I said about clustering, and when I said about PCA, and this is the second edition, which should appear, I hope, finger crossed, by August. I think that is our hope. It is going through the proof stage. And that's all, folks. It's kind of you. And I hope I have not taken too much time of Stephan. Thank you again. So shall I stop sharing?
I guess that would be great, yes.
Okay, done.
Okay, let's see how long it takes till I get my slides on.
Boy, there is a lot of folks.
Okay, yeah, looks like you should see my slides. Excellent. Yeah, so Kanti, thank you very much for the introducing the subject and covering already great parts. I'm also happy that Henrik Wiechers is in the audience. So this is our co-author who has done a lot of work. He'll also be very questions I cannot answer later on. So this is about biomolecules. So I should probably start with those, and they usually comprise a long backbone chain with so-called residues. I think as being talking to a partially biology seminar, I think you're all much better aware than I am. So in DNA and RNA, these are nucleic bases. There are 4 of them. And in proteins, these are amino acids. And here's a human hemoglobin, which is one example of such a long, twisted, and complicated chain. So the DNA sits usually in the nucleus, is less exciting from its structure, but this is transcribed into RNA, which shares in principle the same basis, slight difference, which leaves the nucleus and goes into the rest of the cell and is translated into proteins. Please correct me if I'm wrong, something in the so-called ribosomes. And what is interesting there is that that 3 of such bases correspond to 1 amino acid. And this works like in a zipper. So 3 bases are read off, and then there's a shift of 3 bases. The next amino acid is read off, and so on. This produces a long chain. And so that all these things work, and many more things, it's important that the 3-dimensional structure is as it is. For example, in the hemoglobin, you would have these little pockets transporting oxygen. And if they don't work, then we would all suffocate. Now, viruses can also enter from outside. They have RNA. They also enter from outside and also use ribosomes and produce their ugly and vicious proteins. And now let's go a little more into detail. So here we only do RNA. So this primary structure is called the sequence of these residues. So in our case, this would be just some letters out of 4 letters of the 4-letter alphabet. Sometimes they're bound together in so-called helical structures. This is called the secondary structure, the base bindings. And then things are even more complicated, where part of the backbone is bound together, giving, as you see on the right-hand side, a pretty complicated 3-dimensional structure. And actually, this is the so-called frameshift stimulation element of the coronavirus, which is really vicious, which allows to shift this decoding or this translation just by 1 single base, meaning that in a piece of RNA, the virus can actually encode 2 different proteins. And for this reason, this is of course a target for drug design to make this make it not able to somehow to destroy part of the structure. And by the way, this complicated knot here, these 3 kind of legs are bound together, or helices, is very important for the functionality. But getting to the 3-dimensional structure is quite complicated. So we took this from a paper of Zhang, that's a Nature paper. Those were probably the first to decode or to come up with this 3-dimensional, with a proposal of the 3-dimensional structure in 2021. But they came actually up with several proposals because they're not really sure which it is. And some of the proposals actually contain so-called clashes. We'll come to that. And in particular, they were not able to successfully reproduce these parts. And particularly here at the red arrow, you see that the backbone chain is broken. Okay, before we come to those clashes, let's even take a closer look at RNA. So we have these bases, the residue to a sugar, and here's the backbone. So sugar, phosphor group, backbone, sugar group again, And here, the bond length are pretty much constant, so they don't vary. The angles are also pretty much constant due to laws of chemistry, but the flexibility is given by those dihedral angles, which is a rotation along a central bond. So think of this as a preceding atom, this atom after, and 3 atoms span a plane. So this plane versus that plane, they form an angle, and the angle can actually rotate from 0 to 2 pi. So the folding structure essentially determined by these dihedral angles. Now, how do you get the 3-dimensional structures? So the sequencing is nowadays pretty easy. You get easily the primary structure, the basis. And if you know the basis in principle, or the chemical structure is clear, the only thing you have to find is how the biomolecule is rotated along those dihedral angles. So, and this is pretty costly. People do, for example, electron microscopy. There you find the clouds, point clouds of electron density, like wherever we have the large atoms. There's a mass. There are many electrons. You can do, which is even more expensive, X-ray crystallography, or even more complicated electron nuclear double resonance microscopy, many others. And then people try, often by hand, actually, to fit in the right structure. And this very often causes clashes. So what are clashes? You see this on the right-hand side. So this is from a group, a laboratory at Duke University, that when they reconstruct the molecule, there are these H atoms, and you don't see the electrons because they have surrendered their electrons to the larger atoms. And if you put them back in, you see clashes. That means that the H atoms are reproduced, or are supposed to go at places where which are already occupied by other atoms. And very often these clashes occur within a suite, so between those 2 sugar atoms. As you see here, here's the opposite strand. There are few clashes along the bases, yes, but quite a few clashes along the backbone. So how do people correct for those? One way is to do molecular dynamics. Let the atoms just wiggle along according to the laws of physics and chemistry, which is computationally very costly and works only in approximation and may only find local minima. So you're trying to they're trying to minimize energy. And often you still have clashes. So these methods remove clashes, but very often they do not remove all clashes. Okay, now that's where statistics started. So we've been thinking there should be some statistical idea mimicking, or actually not mimicking, replacing the molecular dynamics. So statistically, you would want to correct clashing suites by similar suites which are not clashing, and you would want to learn them from some clash-free suite. So this leads to classification and clustering. And one simple method is based on PCA, as Kanti said. And for example, to separate well-behaved, this one would be the first PC, and it separates nicely the blue cluster from the other 2, and the other 2 are separated on the second PC. So some 12 years ago, when I was at these famous laser workshops, which were led by Kanti, extremely interesting, those workshops, and he approached me and said, well, how do you do PCA on a torus? And why a torus? Well, a torus is because we've all these dihedral angles, and these many dihedral angles, each lives on an S1, lives on a high-dimensional torus. My first reaction was, well, it should be so difficult. It's a manifold, so just PCA builds on straight lines, replace those straight lines by GD6. Then I took a closer look at those GD6, and so here's the donut. It's the fundamental square of a torus, and it turns out perfectly approximated by a single principal component. Just take an irrational slope. That's a dead end for statistics. There's no residual variation to build any statistics on. Of course, Kanti was aware of this, so he and others had worked on, for example, allowing only few GD6, like the vertical ones, or the horizontal ones, or the diagonal ones. But these are very few. You can't really do PCA building on that. Or people would just do the typical thing for manifolds, just do a tangent space PCA. Okay, but you lose periodicity. Or if you want to keep periodicity, then people came up with building so-called dihedral PCA, which is embedding a single S1 in an R2. This keeps periodicity, but for every dimension, one dimension in torus, you need two dimensions, two real dimensions, which means you need at least two dimensions to model something one-dimensional. So this is also not really the best idea. So I've been discussing with my group, specifically with Benjamin Elsner, who's unfortunately not here, but he's a very important person at this stage or at many stages. So we thought that torus is a vicious space in terms of PCA. What's the most benevolent space with respect to PCA? Well, it turns out, as Kanti already showed, this is a sphere. So if you have data on a sphere, you could approximate it by in an m-dimensional sphere, you could approximate it by an Sm-1 dimensional grade subsphere, just as you see in the picture, project the data on it with respect to spherical distance, then approximate the data by yet another smaller subsphere of dimension m-2, and so on, until you end up at something one-dimensional, which we would call a principal nested circle. It's an S1, the main principal nested circle, and then approximate the data, say, by the best point. This is in contrast to Euclidean PCA. This is a backward scheme, which is different from a forward scheme. If we start with the best point, then the best geodesic passing through it, and so on, because we don't have Pythagoras on the sphere. And then comes the great observation by Sang-Chiu Zhang, who developed this. You could also use small subspheres, as you've seen in the picture by Kanti. If you use small subspheres, you get yet another dimension, more flexibility. Great. Much more flexibility. So here is briefly the difference between PCA and PNS. If you do PCA, you want to approximate your m-dimensional data by the best k-dimensional subspace. It's a small computation that the family of all such k-dimensional subspaces is of dimension m-k times k plus 1. What about if you're on a sphere and want to approximate your data with a k-dimensional small subsphere? Well, this is essentially embed your sphere, m-dimensional sphere in our m plus 1, intersect with a k plus 1 dimensional affine space, gives you a k-dimensional subsphere. So all you have to do is replace m with m plus 1 and here k with k plus 1. So you see, you gain m minus k more dimensions. And this is specifically easily visible if you have, say, if you have 3 clusters, so 3 points can be well approximate or lie on a circle, whereas if you have 3 and you approximate them by a linear PC, then you might not be able to discriminate 2 of the clusters. In principle, you can only perfectly separate 2 clusters with a linear PC. Okay, so it should be a sphere. Let's turn the torus into a sphere. Actually, we need a stratified sphere, so it's the torus to stratified sphere map. And as Kanti said, a torus is defined by angles between 0 and 2 pi. A sphere with polar coordinates has also m angles. There are m-1 angles from 0 to 2 pi. This is the co-latitude. And finally, there's the latitude going around 0 to 2 pi. And with a torus, we have an identification of the zeros with the 2 pi's. This is this tilde. The sphere with an identification of the 0 with a 2 pi. And we also have that these circles collapse at the north and the south pole, so to speak. This is denoted by tilde prime. So what should one do is just half the angles, the first m-1, and the last one can stay as it is. And then we get a new identification, because from the torus we have identified the zeros with the 2 pi's, which means we identified the zeros with the pi's, the north and south poles. So we denote this with double prime tilde. And Kanti has shown this example with if we have a donut in 2 dimensions, we cut it open, then we wrap up those 2 to obtain topologically a sphere, and then we identify north and south pole. So essentially, this is a sphere with north and south pole identified. It's now a stratified space. Same game can be played in higher dimension. Let's just briefly play it in 3 dimensions. So this is a cube corresponding to a 3-dimensional torus. First thing is we halve the first angle and we collapse it. So here we get a line which is identified. Second, we collapse, or we half this next angle, this one. We collapse again for the forward and the backward face. So then we get the north and south pole as before, but now also at the equator, we have this side identified with that side. And the top and the bottom is identified just as being with a sphere as before, because the three-dimensional sphere is the solid sphere inside, where the upper hemisphere is identified with the lower hemisphere. So essentially, we get the m-dimensional torus is mapped to an m-dimensional stratified sphere, which has a manifold part of codimension, or of dimension m-2, of dimension m, and this singular part is a torus of codimension 2. So it starts with m-2, and inside we find a subtorus of yet smaller dimension, and so on. It's a bit like real projective space, only the difference is that real projective space itself is a manifold. This now is stratified, but it's not too difficult to work with. So how would torus PCA then work? Well, we map our data from the torus to the stratified sphere, perform principal nested spheres with the object descriptors as being small spheres as before, but L2 with the squared distance now with respect to the spherical stratified distance. So if a point is near the North Pole, another point near the South Pole, they're actually close by in the stratified sphere. There are a couple of degrees of freedom we also gain, like where should we cut the torus, either at the antipode of the mean, or maybe at the largest data gap. Or how should we order the angles? Should we have maximal distortion and maximal variation of angles inside or outside? So there are essentially 4 combinations of torus PCA. And I should briefly note, this is all part of the concept of the generalized Fréchet means. So for example, all of these small subspheres are minimizers with respect to square distance in some space, in the space of small subspheres, as we've seen of rather high dimension. And the torus mean I talked about is just the classical Fréchet mean. Okay, next step is, once we've gone down to one component, which is a circle, we can actually go a step further for clustering. We can do mode hunting, which has been proposed by Lutz Dümken and Günter Walter for data on a line. So we did the same circle. So in principle, we just order the data. Then we look at the statistics. So these are all differences of data points on the circle. And here we look at the distances between those. So this is a huge number. Because we're in a circle, sometimes we have to take care of the carryover, the 2 pi. But in principle, this is the Dümken-Walter statistic. And then you add up all those guys. And essentially, these are numbers between 0 and 1. If we had a uniform distribution, they would be roughly 1/2. So for a uniform distribution, this would just be some fluctuation around 0. So these are very many data statistics. And now what Lutz Dümken and Günter Walter propose is very simple. You just simulate those guys with a uniform distribution, and you come up with a simultaneous confidence ellipsoid, say with your given confidence level, and then take your data, count the number of violations when you're outside this confidence ellipsoid, for example, and there's a theorem that gives you a lower bound for the number of modes that statistically are present in the data. If you would do some smoothing by, say, a kernel, by a Gaussian kernel on the circle, it would be a wrapped Gaussian kernel. And now the good thing is that the wrapped Gaussian kernel, if we smooth, doesn't produce any new modes. So we can just smooth until, so here's a low bandwidth, we have many modes, until we reach the number of modes we've statistically found. Then we stop and put the cluster boundaries at the minima of our kernel smoothed density. This builds on the Sizer, which was developed by Steve Marin and Shaw Jury, and we developed the Weiser, which is based on the wrapped Gaussian on the circle. Here's another building block. So once we know how to do torus PCA, it might actually help if we look at the data, and this is a typical toy example of the RNA data we're going to encounter. We'll find clusters of varying densities and of varying distances. So we also need to do some pre-clustering before we start our torus PCA. We tried some linkage clustering, average linkage clustering, which finds too many clusters here, can't separate those two, or single linkage clustering, okay, has many outliers, they obviously are dropped. Or we tried our algorithm, and it was designed to find first large clusters, potentially with low density, and then going down, further down, till we find the very concentrated clusters can still separate them. So the secret is to do it adaptively iterative. So we start with a minimal cluster size, which is rather long, or rather high. We compute the linkage cluster tree, and we stop when before the clusters are smaller than S. So rather big clusters at the first instance. We follow the larger branch and store the smaller. And at the end, we also store the larger. And then comes the following. For one iteration, we only take out one single cluster, which is the largest stored cluster. In that case, in the first instance, we would get this cluster. The rest goes back and to the algorithm as the data, and we lower S a little bit to start it again. And then in the second step, we would find this cluster. In the third step, we would be able to separate those two clusters. It's a bit more involved, but I think that's basically the idea of this adaptive iterative linkage clustering, which we call AGE. Okay, now here is what the data looks like. We're going to apply to, this is a benchmark data set of roughly 6,000 suites from the protein data bank, which was analyzed by the Richardson group at Duke University. Here you see 7 dihedral angles. It's a pretty complicated data set. Well, we applied our MINT mode hunting on the main principal nested circle after age, and we found 17 clusters. So far, so good. Are they in any correspondence with clusters found by biological means? Well, we compared to the clusters found in the Richardson group. So our main cluster, which is really large, encompasses 4 clusters of the Richardson group. Good. The second is actually identical. Third one encompasses 2 again. These are identical. So it looks pretty much along the line. We found those clusters that the Richardson group found by more biological reasoning. And here on the right-hand side, you see typical RNA backbones obtained. Well, next step is, now we know how to cluster. How can we statistically do some clash correction? So ideally, what statisticians do is find some correlation, some dependence structure. So we thought we should introduce a meaningful statistical dependence. So looking maybe at a larger scale, not the suite scale, a larger scale. There's these so-called motifs, which are recurrent pseudo-ensemble structures. And we thought that maybe suites neighboring a clashing suite may be less clash affected. Maybe motifs are more stable. And then if that's the case, we would go to kind of motive space, whatever that is, see what the nearest, check for the neighborhood, and somehow project to the nearest non-clashing data, exploiting this multi-scale dependence, because we've done clustering at the microscopic scale. So what's a good mesoscopic scale? It turns out that's here that a half helix turn, these would be 6 sugar centers. Here you see them. So here's our central suite. We look at 2 suites before, 2 suites after. And we modeled this by a size and shape space that was mentioned by Kanti. So it's 6 landmarks in 3 dimensions, giving us essentially 12-dimensional manifold. Upstairs, you see our 7 dihedral angles in our microscopic scale, atomic scale. So these are our 2 spaces. And here's a typical example of a clash, or here you see a clashing suite. This one is a clashing suite. We go to the mesoscopics. So here's the clashing suite. We go to a neighborhood and find that most other clustered or mesoscopic shapes look, they're, and here you see precisely where the big error is. So why should we not, or now let's go back to the suite, space, And here at sweet space, you see that most of those on the right-hand side also belong to a single cluster with a few outliers. So this, obviously, our algorithm goes as follows. Find the dominating cluster in the neighborhood of mesoscopic shape, the dominating sweet cluster. Here's the dominating sweet cluster. Take the mean here, propose this as a corrected sweet. These blue ones are actually the ones that do not belong there. There are only very few. And also over here, correct the sweet, the mesoscopics by taking the Procrustes mean within these ones which belong to the dominating class. There's a unique projection, which is good news. And we also need to, project to the following, that we keep this distance fixed. And also our new suite might have a different length here. So we'll have to project to something that has also the same length here. This projection is unique. Okay, now we did some validation. Is this a good idea? So we went to the roughly 200 within suite backbone to backbone clash suites in the benchmark data. And it turned out most of them had just one single microscopic, one single suite class in there. Most of them had that the first class is dominated by 90%, so 80%. Very few have dominating class that they're just barely dominating, or 50% dominating, but most of them are very strongly dominating, and more than, or almost half them just have one single class. So it seems to be obvious that there is this relationship. What about the size of the correction we did? So we wondered, if you go back, this could look like a pretty drastic correction. Well, it turns out this is roughly within the order of resolution, which is 3 angstrom for this data set. So we looked at the distance, the size and shape distance of the corrected suite with the mesoscopic, with the original one divided by the resolution, and here 3 dimensions and the degrees of freedom. So this was where we're eventually in a 10-dimensional space. And here you see again a histogram, which tells us these numbers are well below, most of them are below 1, meaning we're within resolution order. A little larger, but at 1.2. So this is also good news. So this gives rise to a clean algorithm. So we find the dominating suite class in a mesoscopic neighborhood of a clashing suite. We compute its Frechet torus mean. This is the corrected suite. And we compute its mesoscopic Procrustes mean and uniquely project the size and shape space of the same length with corrected suite length and corrected mesoscopic shape. And the good news about all this is the correction. So now let's go back to this frame shift stimulation element. The data produced by the group by Zhang and al. in this Nature paper. So we took a look at this suite here, which was also all of the 10 reconstructions they proposed are clashing. So here you see the suite shapes, those clashing suite shapes. Well, our method does the following: go to mesoscopic shape space, and so here you see the red clashing mesoscopics. So they're clashing actually here in the center suite, which is this one, and look at the groups of 50 neighbors, local 50 neighbors. Compute the dominating cluster. In the dominating cluster, compute the Procrustes mean, which are these green mesoscopics here. And then go back with a central suite to see what is the torus mean of the dominating cluster. And it turns out for all 10, it's unanimously roughly the same suite. There's much less variation than clashing ones. So all of them give a single corrected suite here, suite shape here, which is clash free. Okay, I think I'm slowly coming to an end. Brief wrap up. I should mention that this also works for so-called polyspheres. These occur in various applications, like, for example, skeletal representations in North Carolina. So essentially, if you have a cross product of spheres of even a varying dimension, you have the same problem with it as with a torus, because if you have some irrational correspondence between here, you can again approximate almost anything with a single PC. Okay, so it's time, I think, for the take-home message. So I think we've introduced some kind of a smart learning. So we didn't do learning with huge amount of data. What we had was 6,000 suites and roughly 200 of them clashing. So it's not too big a data set to learn from. So we need some smart learning. And the smart learning builds on one side on two levels of molecular shape. Small angles are in the atomic scale and landmarks at a mesoscopic scale, and exploiting a strong correlation between them. The next was in order to come up with our clusters, we did torus PCA for the torus data, which it has the big advantage that it can separate 3 clusters with a single dimension, also in higher dimensions, much more powerful, as I told you. And we also came up with an innovative density adaptive iterative pre-clustering tool, which is able to discriminate clusters of varying density and even clusters, very concentrated clusters, which are closer by than the density of the very spread out clusters. And on a benchmark data set, we actually compared our corrections. So we corrected all clash suites. Eraser, which is I think pretty much the state of the art these days, which was started 2013, which is a very complicated tool, corrected only 60%. This may sound great, but I should, of course, we only address within suite backbone to backbone clashes. Eraser addresses all clashes and does many more things. So Eraser is a very complicated tool, very effective tool, but we believe it could be very well possible to use our correction as a new starting point for Eraser, making it much more powerful. Okay, I think at that point, I'd like to stop. Thank you very much.
So thanks a lot for your great presentations. There is a lot of time for questions, so I have many, but please.
Hi, John Kent here. Very nice talk, very interesting. When statisticians do modeling and compare themselves to machine learning people, the statisticians often like to claim that they're bringing knowledge to the problem. So my question to you is how your methods, they seem to me closer to the statistician than closer to artificial intelligence. How do you see it?
Well, Kanti, you want to go first, or should I give a first shot?
You have first shot, and then
I have first shot.
I know John Kent so well what he's thinking, and it will be unfair. I know the answer, but you carry on. I think it's a good question, and you.
Yeah, it's a very good question. So it's, as with all questions that John asks, they require a lot of thinking. So let me briefly, probably as a first answer, I think it's probably the other way around. We've tried to find, we were looking for some knowledge, not using learning methods, but then exploiting those. So we kind of added some intelligence to artificial intelligence. Actually, I would say it's not artificial intelligence. I would say it's artificial gut feeling. So we added some, because I think learning is pretty much like gut feeling. When you learn how to ride a bicycle, it's not intelligence that helps you finally manage how to steer a bicycle. So maybe we added some intelligence to artificial gut feeling.
So it's the best of both worlds, or a synergy between the two approaches.
I guess so, yes.
Excellent. Thank you.
Good. I'm glad he's happy. I had few things because I think at some stage we will have to bring Bayesians in it because their machine learning is quite based on prior, posterior, although we use it indirectly, but not as a model-based. So yeah, there is also a lot of scope. This is the beginning, it's not the end, I think, and also to convince biologists, I think, whether they will buy this or not, because from their point of view, it is a partial answer since we use only sweets, crash sweets, rather than the whole information. So it is convincing biologists, and I'm sure some biologist will come and tell us all rubbish. Is there anyone?
So I have a question, a couple of questions. So I'll ask one. So it's about atomic clashes. So it reminds me a little bit something like what happens in diffusion tensor imaging. So you have sometimes that tracks overlap. So you have different points, different directions, something like that. Well, there you have basically 2 points in the same point, something like that, right? So together with that, it reminds me, singularities. And usually when one tries to resolve that type of problem, increases the dimension. And then works in the higher dimension when there is some sort of resolution of those singularities. And so my question is, would there be any advantage to proceeding that way? To try to remove this sort of singularity by looking at the problem in higher dimensional space, doing your dimensionality reduction or clustering there, and then project back to the base space.
Yeah, let me think about this. If I get this correctly, yeah, I think this. So in principle, this is a very sounds like a very promising, or it's a great tool to do something going to higher dimensions and resolving the problems you have in lower dimensions. I'm a little puzzled because I cannot quite make the connection yet, because we have this problem in 3 dimensions and we have to solve it in 3 dimensions. So I believe that, of course, the clash would disappear if we go into higher dimensions. But in principle, the clash is wrong. So this is not, I think we're not learning from the clash. We're only learning from the clash-free data. So it feels a bit like using something higher dimensional might be helping in clustering the higher dimensional data, or classifying that.
The way in which I connected it to the so the reason for which I connected to the diffusion tensor imaging problem is that I think there it's a little bit simpler. In my view, it's simpler to figure out, in the sense that to me, there you add on a point of the manifold, you add the tangent vectors. So in practice, you work in a bundle, could be the tangent bundle, for example. So in practice, the problem is there is the configuration space and then the velocity space, which is naturally there. And so in practice, it's not cheating, because the natural space there is includes velocities. Now here there are no velocities, but somehow it's reminiscent of that because of the singularity problem. So that's how I see the connection between the two things. Now, I don't know if it makes any sense, especially from biological perspective or other perspective.
Is the singularity? So do you want to remove the singularity?
I want to figure it out if there is a way which is natural for the problem to basically go around that singularity. It's like I have the singularity just covered here, but then I see it from a orthogonal direction to that.
Mm-hmm.
It's like when I see the 2 overlapping vectors, like crossing vectors, then I see that they're crossing because I just see at the tangent, that section there, right? But if I see the full tangent bundle, there is not that issue, because there are two different points on the tangent bundle. So that's what I'm trying to figure out. If there is a way to understand that from that perspective.
So, but would you consider this singularity as a wrong measurement, or is this a valid measurement?
I would hope to consider that a valid measurement.
Yeah, I think that's the big difference.
Right, because in that case, in my case of the diffusion tensor imaging problem, those for me are, I want them to be valid measurements, because they represent two interesting directions in which the water molecules flow. So that needs to be a valid measurement.
Yeah, we're not so benevolent with those measurements. We consider those as, or not we, but the other biologists consider those as faulty measurements.
Got it. Got it.
So one thing which, with your group, we have used only one data, there you can see a particular COVID RNA, but it could be, beyond RNA, it could be in the protein structure, or it could be on membrane, or so, with you biologists, have you come across this kind of problem, or you see some more use than the one example which we have shown? That's clash is a one clear cut, because the atoms overlapping, as you said, can sit on each other, or which are not allowed, I think forbidden. So, but forbidden is a part of the whole game of biochemistry or protein structure. I think it's starting with Ramchandran plot, there is a lot of forbidden areas where you can't go, I think. So I'm just questioning to the audience if they know, other interesting challenges or examples. By challenges, I mean examples where one can try, because whenever clustering comes, of course, a lot of tuning and a lot of playing around. I think it's not like PCA, comparatively speaking. So that different data may give different insight, I think. Anyway, this is the challenge to your group. Help us, statistician, hungry for data.
Taking notes. Other questions? Anybody? A last minute question.
I don't have any questions. I just thought it was fascinating as a healthcare specialist. I was really amazed. I'm really interested to hear more about the dynamic shape modeling, the project that you're working on, the SMEL project. That sounds interesting. Look forward to that.
Awesome.
Thank you.
Excellent.
So sorry, to both of the organizers, to ask. So have you had any talk in past on all this mix-up? Too many of different tools, including shape analysis, is not as common, presumably, with the group, and especially when you come to probabilistic method, of course, in protein structure and various places. So it's becoming more and more statistics rather than black box.
Right. So to answer the question, so the types of direction that this program is following is kind of interdisciplinary. There are people which have stats or math background, people which are more biological background, and sometimes the talks are directed towards some specific technical topic in maybe mathematics or something like that. Some other times are interdisciplinary, and some other times are biological. So the main thing that are touched are shapes or morphology, and the other is biomechanics and locomotion. through either an applied mathematical lens, computational science lens, or you know, a statistical lens. So that's more or less what people have been presenting. Yeah.
Oh,
I think it's the first time we have a seminar series with 2 presenters, but that's good because we have 2 speakers instead of 1, so it's even better.
I think
I would recommend that.
I think to confuse, you know, because I thought at one point I'll be discussant, you know, when I started, and then, you know, when we started thinking more and more, I thought better to give an overview because it is a heterogeneous group. You know, you have got mathematician, statistician, and biologist. So, so pick up a few things and.
Beautiful.
So it is, yeah, it is very exciting that you are doing interdisciplinary. You know, part of our work also normally is very much interdisciplinary, that there is a biologist sitting in background somewhere or sometime in the foreground actually. And when we started this, you know, long time ago into bioinformatics, you know, going back to year, you know, millennium 2000, you know, it started with protein when I didn't know what protein structure means, what amino acids means, and jargon. And yeah, it has been a interesting journey. I think it's a, and you must be doing a lot of learning. I think that's what I found. You know, that one good thing about biologists, they know what they are talking, but for us, sometimes mathematician, it is a gray area. I think trying to be specific is a, you know, has been a challenge always. But yeah, interdisciplinary is the heart of statistics. You know, science, as I had in my first slide. You know, this, you know, what I use, mission statement. I think that we need both science and statistics. One is incomplete without it, or imperfect without that. So that's the slogan we have been using for our leisure meetings, actually. And
Nice.
Well, certainly we found that, at least in, you know, my field in biomechanics, it certainly benefited from the specificity and the clear definitions that bringing math and statistics brings, because the number of times, you know, Alessandro and I have a conversation about that and he asked me what, you know, a term means in biomechanics, and I discovered that we don't, like coordination, for example, defining what is coordination. Everyone uses that word in their everyday life, but what does it mean from a mathematical or statistical perspective? Because there's a lot to be gained from the interdisciplinary work.
Beautiful. So just to maybe, since we had now done the full hour, maybe I will just thank everybody for being here, and especially the speakers. So it was a great presentation, lots of discussion, lots of ideas, and we are really grateful to have had you in our mathematical laws of morphology and biomechanics seminar series. So thanks a lot to everybody for being here, particularly the speakers. Thank you so much.
well, thank you.
Well, thank you.